Okay, I think we can make a start. So welcome everybody to the first set of inaugural lectures of the new academic year. I hope you all managed to get a break over the summer. I'm delighted that we've got two great colleagues who are going to give their inaugural lectures this afternoon. The first colleague is Martina. It's traditional that I say a few words about each colleague before the lecture. I should sort of say it's a massive achievement to be promoted to professor at UCL. I think it's harder than almost any other university, but I'm obviously biased, and it's really a fantastic achievement. So Martina graduated with a first-class honours degree in applied physics at Limerick University. Did her PhD at Imperial in MRI physics. She is, of course, now an internationally recognized neuroimaging researcher, having published very extensively, including in the very top journals. For example, I was interested to see that she was part of the team that conducted the first prospective study of tattoo safety in MRI and was the lead author on the New England Journal publication of that work. She is now, of course, head of physics at the Wellcome Center for Human Neuroimaging. And since 2017, she has led on establishing 3 new research platforms at the center, 2 3T Prisma MRIs and 1 7T. The 7T is UCL's only ultra-high field human MRI system. And Martina was central to the success of the renewal of the Wellcome Center for 10.9 million. And she has been PI and co-PI on many other major research awards. And as I say, she's published very extensively and has a really strong international reputation. She's also active in public engagement activities with many outreach activities across many domains. So it's a real pleasure to invite Martina to give her inaugural lecture. So hopefully Martina is there and ready to go. So over to you.
Yes, Mike, I'm here. Thanks very much for the kind introduction. I'll just share my screen. Is that visible?
Yes, perfect.
Okay, brilliant. Thank you very much. So it's a great pleasure to give this lecture, and thank you very much, Mike, for the kind introduction. Of course, it's a real shame that we can't be there in person together to celebrate the occasion. And in fact, that kind of celebration would be the main appeal for me giving this type of lecture. And naively at the time it was being arranged, had thought that maybe we might be able to be together, but hopefully things to continue to go in the right direction, and it won't be so long before we get to spend some time together and celebrate all of the various missed occasions over the last 18 months or so. So instead of having the conundrum of where we might go after a UCL sponsored glass of wine, I instead got to thinking, what would I actually title the talk? And so really I asked myself the question of what would I talk about, as I couldn't come up with something, smart and witty like people often manage to do. And I thought it boiled down to 2 things really. Protons, which form the fundamental basis of this amazing imaging technique called MRI. But more than rave about how we can turn protons into images, I wanted to actually highlight the various people that I've had the great fortune of interacting with over the course of my career, and in particular in my time at UCL. And so I'd like to take the opportunity to highlight a few of those people over the course of the lecture, but of course there are many others and too numerous even to mention. One never achieves success alone, and really it's always about being part of a team and having the support and help of those around you to really get the best out of yourself. But okay, let's start with the proton side of things. I mean, don't worry, I won't go into too much detail and so on about imaging, but just to give a little overview of what's going on. We start off with the proton, and these protons have properties of mass, charge, and spin. And together, the charge and the spin give us something called a magnetic moment, which when we put it into a strong magnetic field allows us to have a magnetization that we can manipulate. And then the properties of mass and spin give us angular momentum. And this means that in the presence of a magnetic field, we'll start to get this behavior of precession, so rotation about that field. And this is important because then if we were to use a receiver, an antenna of sorts, what we call array coils, then we can pick up that signal as an oscillation because rotating magnetization will induce a current in a coil. So now we have a means of getting a signal non-invasively, and with that we can build up a window into the brain and create very diverse types of images with this technique. So it's in 2 days, in fact, it will be exactly 20 years since, well, a very dreadful event, but also the day that I graduated with a degree in applied physics. I did that, as Mike said, at the University of Limerick in Ireland. And my degree was very industrially focused, so a lot about semiconductor manufacture. And during that time, we got to spend, as part of our degree, a period working in industry, which was a great experience. And as part of my placement, I went to Analog Devices, where we were creating integrated circuits. And I was working as part of one of the teams there. But it was during this time that I read an article in Physics World, which was the height of cool when I was in college, or at least among my peer group. And this article talked about how we could use functional imaging in order to access behaviors and functions within the brain. And I thought that this idea was just really cool and fascinating. And I wondered about how it worked and what the processes involved were. And to me at that time, it seemed like a much more exciting domain to be working in than the manufacture of chips and so on for computers. So I then started to look around on the web to try and understand where people were doing this type of research and how I might be able to get involved and learn some more about it. Unfortunately, there we go. So one of the first places that I came across was the Nathan Klein Institute for Psychiatric Research, which is based in upstate New York in a place called Orangeburg. It was a bit of an odd sort of place to arrive at, considering I was based in Ireland. But I think this was partly due to the fact that I was spending a lot of my summers while I was at university in New York. And so what I did at that time was I reached out to them and I asked if it would be possible to go and visit and learn about the research that they were doing there. And in hindsight now, knowing how busy these people must have been, it was quite phenomenal that they took the time to have me come and see the kind of work that they were doing and learn about the various pieces of research that they were engaged in. It was really important for me because I knew once I had heard about the research that they were involved in, that it was really a domain that I would love to get involved in. They recommended that I get to grips with Bayesian methods if I wanted to work in this domain. And so this was my first book on the topic. And little did I know that down the line, I would end up in a place that had formulated many theories around the Bayesian brain and made all sorts of other important discoveries in that direction. And in fact, even a few years ago, when I was going through old papers in my parents' house in Ireland, I saw that some of those papers that I was reading, or at least trying to read at that time, were written by people who were at the Phil. And so it's quite remarkable to have ended up there after all those years. So once I knew that MRI was the domain that I'd like to pursue after my degree, I started to again look around on the web to see where might I be able to go and do a PhD on the topic. And I was fortunate enough to come across Joe Haenel's group at Hammersmith Hospital, which is one of the campuses at Imperial College in London. And it looked like they were doing some really exciting work. And so I reached out to them. And in fact, I was in New York at the time because it was the summer after my degree. But I came back to Ireland for a wedding. And during that brief window, took the opportunity to fly over to London for the day. And again, considering how busy people are, it's remarkable that Joe was kind enough to take the time to go out to Heathrow to meet me so that we could have a chat in the taxi on the way back to Hammersmith Hospital, because he had to head off to a meeting elsewhere later on in the day. It was a very nice discussion. And afterwards I stayed and I spent the day with the research team, including going for lunch with them, which they got me to pay for, which I thought was a bit odd as I was the student and they were the researchers, but maybe it was a test, I don't know. In MRI, we often have to clear out our pockets, remove all of our money, our wallets and everything so that they can't become projectiles. So that's what they said the real reason was. It's potentially a convenient excuse for us. In any event, whatever it was, I had a very nice day. It really enjoyed the department and the work that they were doing. And so the following October, I started there working on my PhD. They were doing lots of interesting work at the time, particularly in the domain of parallel imaging. And this is one of the key papers that came from the group just before I arrived. And basically it was showing us how we could make MRI a lot more efficient. So typically we'll acquire images from the brain, say from a single spatial location that we would call a slice. And it can be therefore quite a time-consuming process in order to be able to cover the full extent of the brain, particularly if you want to have high resolution. But what they showed is that actually you could acquire data from 2 slices at the same time, and then you could use different antennas that we have around the brain in order to faithfully reconstruct the original images. The thing is, if you acquire these 2 slices at the same time, you've now halved the amount of time it takes to acquire the data that you need. And if you acquire 3 slices at the same time, now it's down to even a third. So this was a very important innovation, and now coupled with additional innovations from many other groups around the world, this technique is available on all systems and now known as multiband or simultaneous multi-slice. And it's become particularly important for imaging techniques that need to acquire lots of data very quickly. So techniques like functional MRI, as well as diffusion MRI. The person that was behind that work was David Larkman, and I learned really a lot from him during my PhD and even since then. And it wasn't just about MR physics, but also about the world and how it works. And I think this photo captures him really well because he can come across a little bit scary, but when you take the time to look and listen, he's got something very interesting to say all the time, and he also cares deeply. But back to the protons, and the reason why MRI is such a powerful technique is because actually within the body, we have a really substantial abundance of protons, most commonly found within water. And these water molecules are really experiencing a kind of a time flux of environmental dependent behavior. So they can be rotating, they might be translating, exchanging magnetization between each other. They could be vibrating or going on some kind of random walk in a diffusion process. But the type of movement and fluctuations that can occur are dictated by the microenvironment. And as such, it means that we have a means of accessing the microstructural environment by changing how we make our measurements. And we see that this happens over many different spatial and temporal scales. So we can tune our measurement to different types, for example, with diffusion MRI, trying to access the barriers that impinge the molecules from moving about, or looking at the molecular dynamics or how they exchange magnetization. And in this way, we can have different sensitivity to different parts of the underlying tissue. These opportunities were discussed very nicely in a recent review by Nikolaus Weisskopf, Siush Mahmadi, and other colleagues. And they showed that within the brain, this means that we can have sensitivity to all sorts of biological features that are relevant both in health and in disease. And combining imaging and modeling is this research field termed in vivo histology, which helps us to characterize these different microenvironments and their functional relevance at ever finer scales, always trying to push to have better and better descriptors. Much of the validation for the modeling occurs by linking and bridging the gap between the in vivo and the ex vivo post-mortem case. And so those samples can also then, the post-mortem samples can also undergo histological evaluation, ideally quantitatively, to characterize different features and relate that back to the MR signals that we obtain. And this can give us information about all sorts of things like the distribution of various cell types, the distribution of fibers, vascularization, and various molecules like iron content, myelination content, and so on. So I first started working and looking at using this type of information, combining measurements with modeling, when I was at Imperial working as a postdoc there. And I was looking to characterize tissue, not in the brain at that point, but looking in different organs like the liver and the heart. So taking the liver as an example, which is the central organ here for those who aren't so used to looking below the neck. What you can see is that the signal that we have in each of the voxels changes when we change the time of our measurement. In particular, the boundaries here show regions where you've got high signal intensity, low signal intensity, and that's alternating over time. And that's because in this case, we have really very different microenvironments for the protons to be sitting in. They might be in a water molecule, or they might actually be attached to some fat within the voxel. And so as the proportion of those changes, so too will our signal. We can plot that over time and see this kind of beating behavior where we have situations where the fat and the water are in phase and we get a bigger signal, so those bright regions in the images, or they might actually oppose each other and be out of phase, in which case we get cancellation and lower signal. And what we can do is we can combine this information with models in order to extract out specific features and quantify at a subvoxel level how much water is there, how much fat is there, and what are their relaxation properties. And this is relevant in a clinical context. And at the time, the clinicians I was working with wanted to look at it in conditions like hepatic steatosis or in cardiac failure, where iron overload would be an important factor. There are certain challenges to imaging in this type of way, so in the body, as opposed to in the brain. But one of the clear advantages is that in this case, we have very distinct environments. And when a proton is in a water molecule, it tends to stay there. When it's in fat, it tends to stay there. And so we're in a regime we would call slow exchange. So this means that the residency time in a given environment is long, and it makes it a little bit easier to distinguish those different microenvironments. And that's not so much the case in the brain. We tend to have quite fast exchange between different environments. And so then our measurement gets a little bit averaged out in effect. We might have a slightly more intermediate case where it becomes a little bit easier, but there are distinct challenges. But before I started to look at that in the context of the brain and try to address those challenges, my partner Saran and I took a break from science and we traveled around the world. It was a really brilliant experience. We spent most of our time in Australia because, of course, we had to have various jobs to pay for the travel that we were doing. But it was a really super time and great experience. We came across all sorts of wildlife in our various travels. And one of the highlights was swimming with wild dolphins off the coast of New Zealand, which is an amazing experience. But then we also saw really diverse landscapes ranging from salt flats to icebergs to volcanoes to waterfalls, and lots of different cities and towns, all of which had their own distinct kind of appeal. We got to do really cool things like sailing on tall ships or climbing to the top of a volcano. Really great experiences that I'm happy to have had. But one of the best things was experiencing all the diverse cultures around the world and getting exposure to those. So although we'd had an absolutely wonderful time, we did end up coming back to London. And after about 5 years away from MRI all told, though spending some time in other research domains, I saw a position advertised at the Wellcome Centre for Human Neuroimaging, the PHIL, and I decided to apply for that, which was a real big slice of fortune for me. When I arrived, I loved the building, the square, and when I went in, I loved the vibe of the centre. Nick Weisskopf was the head of physics at the time and immediately exuded a kindness and intellect that only added to the appeal of the centre. Siwa Mohammadi was also there at the time, and he was a very stimulating guy to have conversations with. Grilled me quite extensively after my interview about the previous work that I had done and asking very in-depth questions, which was stimulating. Then there was Antoine Lutti, who seemed very grumpy on my interview panel, but it turns out he's a really fun-loving guy. And you see him here with Zoltan Nagy, who was an important influence on my arriving at the center. I did get to thinking, putting this slide together, that actually it was very soon after I arrived that they each started to leave. So I hope we don't need to read anything into that, given that we're still great collaborators as well as, I would say, friends. I don't think there's anything to read into. But in any event, as I said, Zoltan was an important influence on me joining the FIL. And this was because after the interview, he took me on a tour of the center. And while, of course, I was super impressed by the various facilities that were there and the setup generally within the center, it was the conversations which spanned both work and our shared love of music that really got me hooked. And over the years since then, he hasn't disappointed at all, mostly because of his exquisite DJ skills. And so he's been the instigator of many fun nights out. And in fact, he even DJs at various conferences and extends into using visuals that even encompass imaging. And I even got the opportunity to DJ at Pasha, thanks to him. And when I say DJ, what I really mean is he told me what button to push, when to push it, but I'll still take it as I've DJ'd at Pasha. So when I joined the center, one of the first projects that I worked on was capitalizing on the sort of skills that I had based on my time at Imperial, where I had been looking at relaxometry. And now I was applying this to better understand inter-individual differences that occur over the course of aging. And this gave me not only an opportunity to build on the skills that I already had, but to learn an awful lot from those around me, and in particular from Nick, including how to conduct a large-scale neuroimaging study using SPM, the software that was developed at the center. But Nick and the team had also been developing an efficient protocol that allowed multiple parameters to be mapped across the whole brain with decent resolution and whole brain coverage, so that we had an array of different metrics that could then be analyzed in this context. And so we also had hypotheses from very focal studies that had been performed ex vivo in histological samples. So for example, characterizing demyelination effects or iron accumulation effects. But here the key difference was now we were getting to ask these questions on a much larger scale, and of course, crucially, asking them in vivo. And so what we found in this study was effects that were consistent with those histological findings. So we did see effects of iron accumulation and we did see effects of demyelination. But of course, the crucial thing is that although this study was a cross-sectional one, the same techniques could be applied to longitudinal analyses or to look at structure and function concurrently. So overcoming some of the limitations of ex vivo post-mortem work. And in general, this approach and research field, which is now quite large, is referred to as in vivo histology using MRI. So we're typically up on the top row here where we have weighted images. So they might be tailored to a particular aspect of the tissue microstructure, for example. But we can use physical models that describe the MR signal to compute distinct quantitative metrics from these data. If we go a step further, we can use biophysical models. And there what we're doing is linking that MR signal right through to features that describe the actual tissue and its microstructure environment. So it might be mapping the myelin distribution or iron content, perhaps details about the fibers in white matter. And one such characteristic is the G ratio, which describes the relative myelination of a fiber. And this is something that I've collaborated with Siawush quite extensively on. And we've looked, for example, at how you can combine different types of imaging metrics So having sensitization to different aspects of the microenvironments, where we have, for example, water within the myelin sheath or water within the interaxonal spaces. And these can be combined in modeling in order to estimate the G ratio. And this can be important because it can help to distinguish between different pathologies, for example, between demyelination effects or axonal degeneration. And where these two conditions might have the same appearance in a diffusion image, for example, or a metric derived from diffusion imaging, then it can appear differentially when we additionally incorporate knowledge about how much myelin is present to form the G ratio, and thereby allow us to distinguish between the two different cases. But it's not just with COISH, it's a really a great characteristic of these ongoing collaborations, even after people leave the center. With Zoltan, we've looked at how we can improve the precision of our calibration data, for example, in order to get better quantitative estimates. And we've also looked at how these measurements that we have can be compared across different sites and even different scanner types. Because of course, these measures are quantitative. That means they're standardized, they have specific units, and therefore that comparison should be possible, which opens up lots of opportunities. Similarly with Antoine, we've continued to work together and often looking at the effect of motion. This can be really problematic for MRI. And as I mentioned at the start, it can take quite some time to get all of the data that we need. And so we become very vulnerable to motion. And it can be difficult for somebody sitting in this or lying rather, in this very unusual environment to be able to stay still for a long time. But we can also develop mitigation strategies. So if we know something about the motion that's occurred, we can try to incorporate that into our analysis to get the best out of the data. And in fact, many current and former members of the PHIL have been working in this domain, creating software for data acquisition, for data processing, and for more sophisticated analyses under the guise of this HMRI framework. And in the long-standing tradition of the PHIL and the Wellcome Center for Human Neuroimaging, this has been about open science and making everything available to others in order to be able to use it. Nick Weisskopf has been a pioneer in this field of in vivo histology, and I had the really great pleasure of working with him for about 3.5 years before he moved on to the Max Planck Institute for Human, Cognitive and Brain Sciences, which is in Leipzig. And he's a director there now and leading the Department of Neurophysics, where they're doing a wide variety of really excellent work. Just a couple of examples of the type of work they have done would be, for example, characterizing superficial white matter and advancing our understanding of how contributors like iron and myelin content are influencing our measured relaxation rates, which is very important to advance the field and make sure we really do understand what it is that we measure. And he's also continued working with people, not just at the center, but more widely within Queen Square. And here's an example of defining a framework for how intracortical manifestation of neurodegeneration might be posed and how that might be probed by the kind of quantitative metrics that we can get. And while this study started looking at healthy volunteers in Leipzig, it's now been extended to look at Huntington's disease in one of the first studies to start on the 7T system that's at our center. And that is ongoing now. During the time that Nick was still at the PHIL, he taught me really a massive amount, and not just about physics, another one of these people who teaches you more than just the science. The kindness and the intellect that I had seen right at the start when I came to interview was always present. But as a leader, he was also an incredibly empowering and inclusive person and an incredible role model for me. He taught me to make sure that I embraced challenges rather than ever thinking of shying away from them. And his evidence-based approach to decision-making was exemplary and something to try to model oneself after. And what he taught me in this comparatively short time was really important, because when he left, I did become his successor as the head of physics. And it was a time when we were applying for a new center award. from Wellcome. And the process was quite involved, lengthy application, site visits, interviewing at Wellcome. And we were seeking support for a 7T MRI system, which is quite a large, complex piece of infrastructure. And so that was informing the strategy going forward. So it was somewhat of an intimidating time to take the reins, but with his continued support and the immense support of many other PIs at the centre, it was something to embrace. In particular, Cathy Price, the centre's director, supportively entrusted me with responsibilities that empowered me. And also working with Eleanor Maguire and Gareth Barnes, who were also on the interview committee that went to Wellcome, was really beneficial. And I learned so much during the period where we very helpfully got grilled by lots of people across ION and faculty, and it really helped us. And the hard work paid off because the Wellcome Centre Award was successful. And although probably most people will know the centre, just a little overview. Basically, we have PIs who are looking at many different aspects of brain function and behavior and how the brain supports those. We're always seeking to pursue clinically transformative applications of neuroimaging in particular, and imaging and modeling are central to that aim. And because it requires quite advanced neuroimaging techniques to be able to support the different investigations that people are conducting, that is why we had 7T as part of our strategy going forward. But of course, the question is, why is that useful and what value is it going to add? So we go back to our little pictorial of the proton. If we have magnetization in a magnetic field, we will have a certain signal. But if we can boost the amplitude of the magnetic field that it's present in, so let's say we go from 3T to 7T, we get a much bigger signal, a much bigger magnetization, sorry. And so with our receiver coils, we can then have a much larger signal induced, which we can trade for higher spatial resolution. And this means that we can start to ask questions in ever finer scales. And this is exactly the type of thing people are seeking to do. So here, characterizing across different laminae within the cortex, functional behaviors. And this is an example of work done by Peter Koch, where he was looking at how prior expectations might evoke differential signals across these different layers in order to progress our understanding of visual perception. I won't talk you through all of the detailed and involved steps that are required in getting such a complex piece of kit into the center, but I'll give you some of the highlights. And one of them was our trip to the Siemens Magnet Technologies factory in Oxford, and you can see me beaming in the center there. This also gives me an opportunity to highlight two people who were really instrumental in getting the system to the center and working in the way that we would expect it to. And that's David Bradbury, who's our lab manager, and Oliver Josephs, who's a long-term member of the physics group and has overseen many installations of MRI systems to the center. And they were with me through numerous meetings, providing expert advice that really did ensure the success of the project. Here I am on the day that the magnet arrived to Queen Square, which was super exciting, but also rather nerve-wracking. Culmination of a lot of work from a lot of people. And then came the moment when this 17-ton magnet was being lifted by a crane up and over our center to find its new home at the rear of the building. And I really love this photo because not only do you see, well, maybe some people are seeing carrots, which I hear a lot, but it's not carrots. It's really the magnet on the end of some pulleys holding it there. And then we have the center behind, and then it highlights our key position with the Queen Square House, home of the Institute of Neurology, and the National Hospital for Neurology and Neurosurgery across the road. So it just shows our integral position there and the impact that this can have, hopefully for everybody. So then we managed to scan our first volunteer on September in 2019. And that was also a little bit of a nerve-wracking moment. We had a lot of onlookers expectantly waiting to see what kind of data we might get, but thankfully it went very well. And straight out of the box, we were getting some nice data. And just one other thing to say, thank you to the team. When I got my promotion to professor, they framed this photo of the magnet arriving. And so now that's up in my office at the Phil. And I really do enjoy looking at it and thank them for that. So what kind of data are we getting from the system? Well, here's an example of a proxy for myelin. And this is the type of data that we can get at a whole brain level with 0.6 millimeter isotropic resolution. So that's an incredibly fine scale. And I really love looking at these movies because every time when it goes through, you start to see different structures, particularly in the brainstem. There's an awful lot of detail there that's really exquisite. So hopefully you can see your favorite nucleus as it scrolls through and that it projects well on Zoom. But of course, one of the points is that we try to get a really multifaceted view of the brain. And so we have different metrics. So here's one that would be taken as a proxy for iron. And you see that really it's quite distinct contrast and different structures that are highlighted in this type of image. If I put the two together, then you can really appreciate the differences that exist across the two. Of course, in addition to the quantitative imaging, then there is also functional MRI, where again, people are trying to push toward the laminar level, and particularly to be able to distinguish feed forward and feedback mechanisms that are occurring within the cortex. These data are acquired at 0.8 millimeter resolution, but still take less than 4 seconds to acquire. And in fact, a lot of the development that's happening at the moment is in the domain of fMRI. People want to ask all sorts of questions like, how do expectations shape perception? What brain regions are involved in autobiographical memory formation and how do they communicate with each other? And how do these conditions go, or how do these links go awry in disorders or in disease? And it's people like Peter Koch and Ellen McGuire who are really pushing that laminar fMRI program. And they don't give us an easy time, I gotta say. They always want to look in the more difficult regions, and every time I think we've reached the most difficult, they push it that little bit further. But it's exactly the type of thing that makes it so valuable to work at Queen Square, because you always have people who are trying to push through the boundaries and progress to the next step, and not having, you know, technology stand in the way, let's say. So we welcome these challenges and these collaborations, but they really require that you have a great team. And in fact, we're lucky on that front too, because the physics team is filled with talented individuals. I was particularly lucky that Nadège Corbin was the first person I recruited when I became head of physics. And if I had to sum her up in one word, it would be success. She's a phenomenal talent, creative, helpful, collaborative, always seeking to enable other people and their projects and the kind of difficult things that they want to do. She's always, or nearly always, smiling and approaching things with a really positive attitude. But beyond her academic abilities, she also was the person who introduced me to the Queen's Larder, which does seem a bit ironic given that I was there 3.5 years before her. But in my defense, I had more typically hung out with people who were based at Queen Square House and had different haunts. But it was good times, many, many good times. And she also pushed me into doing more active things like taking up running, which I don't think I would have done if she hadn't picked my competitive streak. And we each pushed each other, although she was generally a lot faster than I was. But we got to use this in a positive way. And here we are after a 10K run that we did, where we also managed to raise some money for charity. So that felt good, even if the sore legs didn't. Despite her return to France, I'm really happy to say that we continue to collaborate, and we also are now really good friends, I would say. And as a nice swan song before she left about a year ago now, she managed to have the excellent achievement of producing the first paper that was based on work done on the 7T system that we had in place. And so what we were trying to do here is understand the impact of a common simplification that we make and how it might affect our measurements, and in particular, how it might be different between 3T and 7T, and how we could mitigate it for the studies that we're seeking to start on the system and are now using these techniques. As I said, one of the great things about the FIL is that you do continue to collaborate with people even after they've left. But actually, it also is the case that you don't even need to have overlapped with them, such as the way that the network evolves. One example is Patrick Freund here, who I met when I was doing the aging study, even though he had already left the FIL, but he was had spent time there and was a regular visitor. And so we started to additionally look at aging effects within the cord, the spinal cord, which he's particularly interested in. And he's now based at the Balgras Spinal Cord Imaging Center in Zurich. And so together we have also managed to have a consortium established to look at extending spinal cord imaging. And that also involved Nick Weisskopf in Leipzig and Siush Momadi in Hamburg, who are also in the photo there. And actually this is from the Kickstart meeting for that particular grant. And in that grant, we managed to recruit into the physics group, Yale Balbastra, who was at that time in John Ashburner's computational neuroanatomy group. And he was a really fabulous link between the methods and the physics teams. And from that, a lot of really interesting collaborations emerged. For example, the analysis of quantitative data could be extended within SPM to cover not only the brain, but additionally the cord. But through further collaboration with John's group, we showed that a generative modeling approach could be used to determine missing values in the quantitative imaging sense. So all of the voxels that are within the little dashed green box have been determined based on the values outside of the box. And this is a really powerful approach. As I highlighted just a little bit before, motion can be a really big problem for us. So you can get these kind of rings present in the image when somebody moves during an acquisition. But actually, if we use that framework for a generative modeling of the data, then we can infer what those values would have been in the areas that are corrupted by the motion. And you see that it does a really great job of reducing, if not completely removing, the artifacts that were present due to motion. We can also look at lots of interesting work that's happening from the newer members of the team. And as an example, we often assume that the relaxation rate or the behavior of the magnetization over time is consistent, even if the signal amplitude itself might change initially based on particular preparations. So we describe it by a specific parameter. But actually, owing to the fact that we really do have these differential microenvironments present, the water that might be in a myelin environment versus in an intraaxonal space or an extraaxonal space, actually we can find that these measures vary depending on measurement parameters that we don't ordinarily assume that they do. So for example, based on the flip angle that we use for our excitation, so to generate the magnetization, we might obtain one map for this particular parameter. But if we change that parameter of the flip angle, then we get a different one. So this could be seen as introducing a confound or a bias to our measurements. But in fact, we can turn things on its head with appropriate modeling. And what we can do instead, just to show you some particular regions, for example, the corpus callosum, where this is particularly obvious, or indeed in superficial white matter. Now, what we could do is get something that's independent of the flip angle. So we have a more replicable and consistent measure. But we could also use the modeling to try to determine how much myelin water there was in each and every voxel. This is work that's being done by Georgia Malota, who is a fairly recent recruit to the physics team, joining during the pandemic, so about a year ago. You could argue that that's a difficult time to be joining, and there are quite a few members of the team who have joined like that. But as we start to get back to the center, we can start to not only do our work together, but have some fun together. The other person you see here is Vahid Malekian, who also joined within the last year. He's doing a lot of work to try and mitigate artifacts that we see in functional imaging. And he's doing a really great job there. He's also doing a really great job at ping pong, which I don't particularly like. And I wonder what happened to letting the boss win, but it certainly doesn't happen in the physics group. So another collaboration across Queen Square that I wanted to highlight is this one here. And actually, it turns out that the impact that time has on the MRI signal is not so dissimilar to the impact that the size of the GA1 repeat has on the age of disease onset in Huntington's disease. And indeed, just like we find that changing a flip angle might change the rate of our signal decay, similarly, it seems that having a particular interruption and where that interruption occurs will also change the impact of the repeat size. And so that means that we can use very similar modeling in both contexts. And this was a particularly important collaboration for me because the lead on it was Saran Netesinger, who's from the Department of Clinical Neuroscience and Movement. And actually, I met Saran on the very first day of my PhD nearly 20 years ago. And although we've had the opportunity to acknowledge each other in various papers over the years, this is actually the first time that we got to publish as co-authors. So it was a very nice, somewhat of a product of the lockdown and the pandemic, and a very nice achievement for us. Another person I acknowledge in every paper I write is my little sister. She's the reason behind the F in the F initial that I use in my academic output. And I do this because she has shaped me more than anyone in life. And therefore, everything that I achieve is also a credit to her. And of course, to the rest of my family, who I'd also like to thank. And I'd like to end by thanking all of you for listening to me and indulging in the story of my academic career and how I got to where I am today. A final thank you to all those who have supported me over the course of my career. In particular, all of the amazing people at the Wellcome Center for Human Neuroimaging. Thanks very much.
Thanks, Martina, for a really fabulous lecture and telling us about Pasha. That was interesting. So I'd now like to ask Cathy Price if she'd like to give a vote of thanks. Cathy, are you there?
Hello.
Yeah, go ahead.
Okay, so thank you, Martina, for putting the fun into physics and for so clearly explaining your scientific agenda and the power and potential of MRI. What is so remarkable is that your scientific agenda is only a fraction of what you do. In your role of head of physics, you're actively involved in supporting all new MRI applications, continually problem solving to ensure that we facilitate the most advanced MRI programs. You are the most invaluable deputy head of department, the master of developing our standard operating procedures, and a fabulous role model to all. And without you, there would be no 70 scanner at Queen Square, because it was Martina who convinced the leadership team that we needed a 70 scanner. It was Martina who wrote all the persuasive texts in our 70 fMRI funding applications. And you're a brilliant communicator and socializer, as we've seen in your slides, always the last of the dance floor at the office parties, beating everybody at fuzzball, even if you don't beat people at ping pong, and doing the occasional 10K run. And of course, supporting the Irish rugby team. We're very lucky to have you at UCL, and it's my honor to thank you on behalf of the department and the Institute for your commitment and support to all of your dedication to clinical neuroscience. So thank you, Martina.
That's great. Thanks, Cathy. I'm sure we all echo those sentiments. So thank you for a great lecture, Martina.